wonderful students. I, Dr. Jyoti Vaipavi, welcome you to today's lecture video, that is microcirculation. These are the specific learning objectives of today's lecture. At the end of this lecture video, students are expected to have a good knowledge about these topics. So please go through them properly. So, what is the purpose of circulation? What do you think? Right, mainly for the transport of nutrients and oxygen, to removal of waste products. If you give a thought in the entire circulatory system, which is that part which serves the purpose of circulation? Yes, exactly. It is the capillaries. So we can say that the most purposeful function of circulation occurs in the microcirculation. Well. What is this picture doing here? Wondering what footballer and a football stadium has to do with the microcirculation? Well, there's something very interesting. The peripheral circulation of the whole body, if you take out the peripheral circulation, it has about 10 billion capillaries. Yeah. If you take out all of the capillaries from a human body, if you are able to do that and spread out on a surface area, it is estimated to be around 700 square meters. That is around 1/8 of a football stadium. Amazing, isn't it? And one more very fascinating thing is that you take any functional cell in the body, it is not more than 20 to 30 meters away from a capillary. So, this is how extensive our capillary is arranged in our body. So, with this amazing fact, let's learn the structure of microcirculation. Well, before actually going into the structure of microcirculation, we shall look at this illustrated diagram over here. Okay. See, this is showing the circulatory system. All the deoxygenated blood is coming to the right atrium, from there right ventricle, from here pulmonary artery, deoxygenated blood is going to the lung for exchange. This part, this capillary system here is called as the pulmonary capillaries. So this part of the pulmonary circulation has this pulmonary capillary bed. Here exchange happens, oxygenated blood comes back to the left atrium, left ventricle, aorta, arteries. Again it divides into arterioles and systemic capillaries. Here for just simplicity purpose we have just showed two tissues over here, but like this it goes to the entire body. The oxygenated blood and important nutrients are supplied, again so on and so forth. So why this picture here? To tell you people that there are two types of circulation, pulmonary and systemic, which you already know. And also you have to remember the capillaries are not just present in the systemic, in the body, but also here pulmonary capillaries. Okay, so with this we'll move on to the components. So what constitutes the microcirculation? If you take any organ, there is a nutrient artery for that. Each nutrient artery entering an organ, each nutrient artery entering an organ branches 6 to 8 times when they become small enough to be called as the arterioles. Further, this arteriole branches again 2 to 5 times again to form the meta-arterioles. The point where meta-arterioles continues as capillaries, at this point there is the smooth muscle covering. Smooth muscle encircles this meta-arteriole here. This is called as the precapillary sphincter. Okay, this precapillary sphincter controls the opening and closing of the capillaries. From here venules are formed. Capillaries come through this and then they form together to form the venules. These are the main components of the microcirculation. So if you can put it in a flow chart, so the components are like this: arterioles, meta-arterioles, precapillary sphincter, capillary and venule. And most important thing here is what we should not forget, is this lymphatics, right? They play a very important role in microcirculation. So, a good student always remembers microcirculation. It's not just this, but also constitutes the terminal lymphatics. This picture shows the components of the microcirculation along with the lymphatics. It emphasizes the importance of lymphatics in the microcirculation. So you can see this, it's again a schematic diagram just showing the right and left ventricle, pulmonary capillaries and systemic capillaries. So whatever the fluid is coming through these capillaries are filtered into the interstitium, and some of them is again reabsorbed into the venule. Some fluid and some substances which cannot be reabsorbed, uh, it cannot remain in the interstitium. So there the function of lymphatic starts. It helps in bringing back all the substances, all the fluids back to the venous system. It does not allow the fluid to accumulate in the interstitium. It also plays a role in managing the fluid homeostasis of the interstitium. So let's now learn the structure of capillary wall. So this is again a schematic diagram showing a capillary wall. Capillaries are made up of unicellular layer of endothelial cells, which is surrounded by thin basement membrane over here, okay, on the outside of the capillary. Now, tell me one thing. What do you think? There is this endothelial cells and basement membranes. What is it which is more permeable? Is the endothelial cells more permeable or the basement membrane? Which is more permeable? So let's think. Basement membrane is also called as extracellular matrix, and it is actually made up of network of collagen, isn't it? So they are very porous. They do not control anything. It's the endothelial cells which acts as the real barriers, which controls what has to be filtered, what has to be reabsorbed. So all the function of the microcirculation is mainly happening in the endothelial cells. So I have put this picture to show you how a ultra microscopic picture of a capillary bed looks like. So capillary walls are just 0.5 micrometer. They're made up of endothelial cells. The diameter of the capillaries is about 4 to 9 micrometers. As you know, these are the red blood cells over here. Can you see? As we all know, red blood cells is 7.2 micrometer thick in diameter. So you can see this, the the RBCs are in the file of RBCs are just going one after the other. They cannot make two rows. They're just squeezing through this capillaries. So I just have put this picture so that you remember whenever I say a capillary, you should remember how thin they are, how extensive they are, and how RBCs are squeezing through them. Okay, so this is how the structure of capillary wall looks like. If you take the human systemic circulation, some capillary beds are very leaky, like glomerular capillaries, right? They're all the time filtering. There is filtration happening, and some are not at all leaky. There is nothing passing through them. That is the blood-brain barrier. You know why this disparity is there? Differentiation is there. It's mainly because of the pores in the capillaries. What is it called as? The pores. Now, where are these pores present? Pores are nothing but interendothelial gaps present between the two endothelium. Okay, so they're called as pores or interendothelial gaps. Based on whether gaps are there or not, the capillaries are classified as continuous, fenestrated, or sinusoidal. Among this, the most common, extensively found in the entire body is the continuous. Especially, it is characterized by basement membrane is intact, no gaps in between the two endothelial cells. It is just the tight junction, nothing passes through. Okay, so this kind of continuous capillaries we find it in blood-brain barrier, also the muscle cells, skin, all that. So coming to the fenestrated capillaries, it is one of that, fenestration means, what do you mean by fenestration? If you can see the endothelial cells here, there are many pores. There are holes in the endothelial cells, you can say like that. So this makes the capillary more permeable and larger molecules can pass through this. The number of fenestration and their degree of permeability vary according to the locations. Okay, it's mainly found in the small intestines, where the nutrients has to be absorbed, as well as in kidney, which filter the blood all the time. So we find the fenestrated capillaries. Then the next type is the sinusoidal capillary. It is the least common type of capillary but most filtered. So sinusoidal capillaries has large gaps in between two endothelial cells. There are more gaps. And when you look at the basement membrane, it is also not continuous. It is broken. It looks like a Swiss cheese, right? Where are these found? They're mainly found in the bone marrows. The bone marrow forms new blood cells, isn't it? The cells must enter the blood supply and can only do so if there is large opening of the sinusoidal capillary. They cannot pass through the small openings either here or here. So bone marrow, we can find the sinusoidal capillaries, and also the liver. Liver also requires extensive specialized sinusoid capillaries in order to process the materials brought to it by the hepatic portal vein from both digestive tract as well as spleen, and to release plasma proteins into the circulation. So these are the types of capillaries we found based on the pores and the intactness of the basement membrane in the body. So if you can see the leakiness or the filtration capacity, the continuous capillaries are nothing can pass through it. It's very tight. So it's difficult for the substances to move through them. Whereas the discontinuous, that is the sinusoidal, they're very leaky, fenestrated, are in between. Well. What is this? This vasomotion is on and off. Okay. Vasomotion means the blood is flowing in the capillaries and are not continuous. They flow intermittently on and off, just like this. So every few seconds they on and they are off. This phenomenon is called as vasomotion. The blood flow in the capillaries are intermittent. This is called as vasomotion. Okay, now we have to learn. So what controls this on and off? Why is this on and off movement? As we all know, there are precapillary sphincters. They can close and open, okay, according to the oxygen supply of the tissue. So what do you mean by according to the oxygen supply of the tissue? For example, if you're jogging and your calf muscles require more oxygen, so in this tissue, the capillaries, the precapillary sphincters open more often, open, and also they're open for a longer time, so that they get more oxygen, because oxygen consumption is more in this particular tissue. Precapillary sphincters, they open for a longer time and open more often. So this is how, based on the oxygen supply, the vasomotion of is regulated. Okay, now coming to transcapillary exchange. Okay, so blood is coming to the capillary. Some substances are filtered, we know that, and some substances are reabsorbed, we know that. Which substance and some substances remain in the capillaries itself. It's not filtered, it's not absorbed, it's just there in the blood. So what is that? Which is, what is all this? Why is all this? We will understand all these under transcapillary exchange. Why some substances are filtered, why are not filtered, and all that. So transcapillary exchange takes place by one of these processes, either through diffusion, vesicular transport, or filtration and absorption based on forces present in the capillaries. Okay, so here is the schematic diagram. Over here is the endothelium. This is interendothelial space. Some of the substances are coming here, some of the substances are just coming through the endothelial cells. Okay, if you take a cross section as depicted in this of a capillary wall, this is the cross section. So this is the plasma, these are the endothelial cells, and these are the interstitial fluid. If you can take this, so small water soluble substances, see, like sodium, potassium, and amino acids, the exchange between plasma and interstitial fluid by passing through these water filled pores, sometimes through the spaces, or there is water pore channels in these endothelial cells which transfers these substances. Whereas lipid soluble substances like oxygen, carbon dioxide, they just pass through the endothelial cells through passive transport called as diffusion. And some molecules, some proteins, which has to be crossed from here to here. For example, if you take um, pancreas, the insulin which is produced, it has to be transported. How does it transport? Or any protein molecule which has to be transported. So protein peptides which has to be transported, is transported via vesicular transport. And what about this? Big plasma proteins, they cannot be transported, they just remain there. So this is a rough idea to tell you how transcapillary exchange occurs based on the solubility and also the molecular size. So by far the most important means by which substances are transferred between plasma and interstitial fluid is diffusion. As we all know, it is a passive transport. So, like everywhere, transcapillary diffusion also follows Fick's law of diffusion. So, microvascular diffusion is directly proportional to the concentration gradient, surface area, pore size, lipid solubility, velocity, temperature, whereas it is inversely proportional to the molecular size and thickness. So, just like all diffusions, so all the lipid soluble molecules just diffuse through the transcytosis. Proteins like peptide hormones to be moved across are exchanged by vesicular transport. Here you can see the capillary endothelium. It's a little bit enlarged to show you the how the molecules, how the protein peptides are transported, because they cannot be undergo diffusion or water soluble transport. They are transported across the endothelium by vesicular transport. Just like how when insulin is produced, it is transported to the circulation via vesicular transport with the help of cytoskeleton and the capillaries here. So the second means by which exchange is accomplished across capillary walls is bulk flow. A volume of protein-free plasma actually filters out of the capillary, mixes with the surrounding interstitial fluid, and then is reabsorbed. So this process is called as bulk flow because the various constituents of the fluid, not only the water but also the solutes, are moving in bulk or as a unit, in contrast to discrete diffusion of individual solutes down the concentration gradient. So this is one of the major uh, part, like how the transcapillary exchange takes place. If you can see this picture, the fluid leaving the capillaries at the arterial end, this part is called as the filtration, and the fluid reabsorbing back into the venous end, it's called as the reabsorption. The movement of fluid and solutes from this arterial end, filtering and again reabsorbing, this is called as bulk flow, or the major process of filtration and absorption. Okay, so you have to know what is that force which is acting on the capillary, what is that force which helps in this bulk flow, why is this movement happening, so much of water and solute movement is happening, what is that force, that force which helps in all this filtration and absorption in this bulk flow is called as the Starling's forces. What force? Starling's forces. So you should have to remember this. So we come to the end of this lecture. So to summarize, in today's lecture, we have learned the functions of microcirculation, we learned about the components, we learned about the structure of the capillary wall, we learned there were pores, and based on that we have types of capillaries. We understood what is vasomotion and how is it regulated. We understood different processes of transcapillary exchange like diffusion, vesicular transport. Also, we understood the bulk flow, the filtration and absorption, and the force acting on it, force which helps in filtration and absorption. So, children, next class we shall be discussing forces, Starling forces, and edema. Please go through the video again and refer Student Kaitan Book of Physiology for better understanding. Thank you all for a patient watching.